So today, we're going to talk about something pretty incredible, decoding the language of life itself. We're diving into a technique that lets scientists read the genetic instructions tucked inside every living thing. It's how we figure out what makes all of us unique, and it all starts with a simple comparison. Okay, so here's a big question for you. How do we really know a human is more closely related to a chimp than to a mouse? I mean, it feels obvious, right? We kind of just know this, but how do we actually prove it scientifically? How can we put a number on that relationship? Well, the answer is hidden in the source code of life.
The way scientists tackle this is with a really powerful process. By comparing these long strings of DNA or protein, they can start to see these deep connections. They can figure out what certain genes do, they can predict the 3D shape of molecules, and yep, you guessed it, they can trace back the branches of the evolutionary tree.
So what is this powerful technique called? It's called sequence alignment. And honestly, the best way to think about it is like life's ultimate spell check, or maybe its ultimate comparison tool.
Okay, so the textbook definition is pretty straightforward. It's a method for arranging sequences, DNA, RNA, proteins, you name it, to find spots where they're similar. Basically, the whole goal is to just see where things line up and, where they've gone their separate ways over time.
This is my favorite analogy for it. Imagine the entire genetic code of two different species. Think of them as two giant Word documents. Sequence alignment, it's literally like putting them side by side and hitting track changes. It shows us every single edit, every mutation, every insertion, every deletion that has happened over millions of years. And this is what it looks like in practice, a really simple one anyway. You've got sequence A on top, B on the bottom. See where the letters, the bases match up? But look closer. See those dashes? We call those gaps. They're just placeholders, showing where a letter probably got added or deleted in one sequence, but not the other. It's a visual map of their shared history.
Now, as you can probably guess, there isn't just one single way to do this. Scientists have a couple of major methods in their toolkit, and they each have a different job to do. On one hand, you've got pairwise alignment. Pretty simple, right? It just compares two sequences at a time. On the other hand, there's multiple sequence alignment, which is a beast. It tackles 3, 4, even hundreds of sequences at once. Now that's amazing for finding patterns across a whole bunch of species, but it gets complicated fast. So for today, we're going to stick to the fundamentals, the real building block of it all, pairwise alignment. All right, so that leads us to the next big question, right? If you're only comparing two sequences, with all the different ways you could line them up, putting gaps here or there, how on earth does a computer figure out the best alignment, the one that's most likely to be biologically real?
Well, the answer really boils down to two main strategies, two ideas that are basically the foundation of bioinformatics: global and local alignment.
Okay, so global alignment. You can think of that as the big picture approach. It uses something called the Needleman-Wunsch method, and it tries to match the entire length of both sequences from start to finish. It kind of assumes they're pretty similar overall. But then you have local alignment. Using the Smith-Waterman method, it does the exact opposite. It doesn't care about the whole sequence. It's on a mission to hunt for the single best little chunk of similarity, even if that chunk is hidden inside two sequences that look totally different otherwise.
Let's stick with an analogy. Think about global alignment like this. You're a professor with two student essays on the same topic. You're comparing them from the very first word to the very last one, trying to get an overall similarity score. You care about the whole document.
And how does it work? Well, it's basically a three-step dance. First, the computer builds a 2D grid. We call it a matrix. Second, it figures out the best path by tracing backward from the bottom right corner. And finally, it uses that path to build the alignment that we actually see at the end. So, this grid, this is how the computer keeps score for every possible match. Now, we're not going to get bogged down in the math, but here's the absolute key for global alignment. The algorithm always starts tracing back from the very end, that bottom right corner. And by doing that, it forces the solution to stretch across the entire length of both sequences.
All right, now let's switch gears to local alignment. The analogy here is pretty cool. Imagine you're searching through two completely different books, say Moby Dick and a modern biology textbook. Your job is to find one identical, powerful paragraph that just happens to be in both. You're going to ignore everything else and just zoom in on that one little island of similarity.
And the process, it looks almost the same, right? Create a matrix, trace back, build the alignment. But, and this is a huge but, where you start that traceback changes absolutely everything.
Okay, so here it is. This is the critical difference.
Instead of being forced to start in the corner, the local algorithm hunts for the single highest score anywhere in that grid. See that number 8 right there? That's our winner. It starts the traceback from there and, this is key, it stops the second the score drops to 0. That's how it so perfectly isolates that one best paragraph and just throws away all the noise around it.
So, okay, we've got these really clever computer tools, global, local, but why? Why does any of this matter? Well, because this whole process lets us unlock some of the deepest, most fundamental secrets of biology.
This is how we answer that very first question about humans and chimps. It's how we can look at an unknown protein and make a really good guess about what it does. It's how we find a tiny single-letter mutation in a gene that might cause a disease. I mean, in modern biology and medicine, this stuff isn't just a nice-to-have, it's absolutely essential.
And its impact is just massive. You can see it's a total cornerstone for evolutionary biology. It's absolutely critical for discovering new drugs and medicine. And it's vital for functional genomics, which is basically the field trying to figure out what all of our genes actually do.
So from tracing our ancient past all the way to designing the medicines of tomorrow, sequence alignment is really our microscope for reading the book of life. And that kind of leaves you with one final big thought, doesn't it? With trillions and trillions of pages of genetic code still out there to explore, what other incredible secrets are just sitting there waiting to be aligned?